I'm Dr. Neeraj Khatri Sapkota, and today I'm going to talk about the mechanism of red blood cell formation or the process of red blood cell formation, and which is known as erythropoiesis. Let's write erythropoiesis. So, erythropoiesis is Poiesis. So erythropoiesis is the process of formation of RBC. So RBC is going to form, right? So RBC, if you see intrinsically the RBC content, then RBC is formed by some of the proteins that surrounds the RBC cell membranes that has some sort of band, and that is said to be spectrin protein, right? And this RBC cell, inside the RBC cell there is a molecules which has very high level of significance of functionality, and that is known as hemoglobins. Each RBC cell contains about 4 molecules of globin chain, 4 chain of globin, and these globin chain are named as alpha, 2 alpha and 2 beta. Though there are some dissimilarity or there are some changes that occurs in the RBC of the fetus hemoglobin and the RBC of the adult hemoglobin. So this hemoglobin molecules is going to be synthesized during the process of erythropoiesis, and this erythropoiesis process takes place in the bone marrow in the adult, and the bone marrow contains some of the progenitor or stem cells, which is known as hemocytoblast cell. Network of cell is there, and this reticular cell, and this hemocytoblast cell is even known as hematopoietic stem cells, and this hematopoietic stem cells is from the myeloid, myeloid series, right? Myeloid series or myeloid lineage, right? Whereas another lineage of network of hemocytoblast progenitor cells comes from lymphoid lineage. So there are two lineages. One is lymphoid, and this is lineage, right? So lymphoid lineage gives you lymphocyte cells such as T cells and B cells, whereas this lymphoid lineage is giving you T cells and B cell, whereas myeloid lineage is going to give you the erythromyelocyte cells. We can say it is going to give you erythrocyte cells, and it is going to give you some of the WBC cells. So this WBC cells is coming from myeloid series, and this T cells and B cells is going to come from the lymphoid lineage series. So this network of cells that is present within the bone marrow can have some rate of formation. Rate is the determinant of speed. So how fast the RBC is going to form is determined by the presence of reticulocyte cells in the peripheral blood. So reticulocyte cell is the immature or completely not formed, incompletely formed mature RBC, which later on get converted into RBC. So that is the part of the erythropoiesis handling. And this erythropoiesis process requires RBC with some of the protein synthesis required there. The most part is RBC is now going to replace by the hemoglobin. Initially it contains, for example, in the proerythroblast cell or in different stages of erythroblast cell of this myeloid series, it contains nucleus, and now this nucleus is going to be shed out from the RBC, and that is going to be replaced by the hemoglobin. So incorporation of hemoglobin takes place in about the different stages of the erythroblast cell and changing it into the normoblast cell. When it reaches to the reticulocyte stage, which takes about, let's say, 4 days, then about 80% of the nucleus is getting replaced by or got reflexed by the hemoglobin. So reticulocytes count give us an idea of rate of formation of the RBC. That means the process of RBC. So with this, we have understood what we need for the formation of RBC. That is the process of erythropoiesis. In second class, the next class, we will study